Besides gene duplication, I also want to talk about genome duplication. So genome duplication now is very obvious. You often heard about which species is a tetraploid and which species is originate from some time ago, and in this lineage the genome has duplicate like twice. Okay, but how in early days how scientists recognize genome duplication is by the cytology. So in early 1910s, people already recognized several plant species as huge genome by counting their chromosome. So the experiment there was relatively simple. We examined the karyotype of the plants, and we can compare like the karyotype among close related plants and ask whether they have same number of chromosomes and what's the chromosomal changes among or between species. So that was a very easy psychological study, and we still use this technology today. But in that early years, without like many molecular biology and DNA knowledge, and people already recognize plant species are sort of ancient tetraploid. When we say ancient tetraploid, is when we compare, for example here, maize with its close related species, we recognize the genome is doubled, so we say it's a tetraploid, but the genome duplication has occurred long time ago during the separation of the two species. So we say it's an ancient tetraploid species. And because we can recognize, we can count the chromosome number, and we can compare the chromosome shape cytologically. So by doing that, we can recognize a species as a tetraploid, compare its close related species. And in speciation, this is really important because once a tetraploid is formed, and immediately it's different from its ancestral population, ancestral species, because showing on this slide is the karyotype of the two species and also their gametes. And you can see if the two gametes form, one is the haploid genome, the other is the haploid of the tetraploid. So actually it's the diploid genome based on the original species. Then if the two gametes fertilized, then you will see like a 3-end individual, and which will be problematic when this individual perform meiosis and form gametes. So this is one of the interesting topics in speciation, because we know once the tetraploid form, the reproductive isolation complete immediately. So we know there are two different types of whole genome duplication, or have like different ploidies. So one is autopolyploid. So polyploid is like a multiple duplication. It can be tetraploid or even more duplication, whole genome duplication. But the autopolyploid means the duplication events only involve in one single species. Showing in this slide, you know, when the cell begin to entering meiosis, the chromosome needs to be duplicate, right? So replicate, and you have a 4-end stage, and they would go 2 rounds of division and form the haploid. So in this process, if the mutation occur that leads to whole genome duplication, then the gametes form sort of an individual and produce offspring. Then in this case, we'll have an autopolyploid species. So So tetraploid is one of the simple example based on this one. The other one is allopolyploid. Allopolyploid involve at least 2 different species as showing on this slide. So this slide showing there are 2 different species, and this is just one of the possible ways to form a allopolyploid individual. So there are different ways to sort of produce very similar case. To illustrate the chromosome, we use different color. So one is blue, the other is red. And in B species, they have 2n equal to 4, and A species 2n equal to 6. And of course, there's meiosis and also fertilization. But at the end, you can see the final product after the whole genome duplication. You will see the allopolyploid individual contain the genome from both species. So they contain 2n equals 10. So there are 2 sets of chromosomes from species B and 3 sets of the chromosomes from species A. So when this type of genome duplication involves or contains more than one species' genome, and we call it allopolyploid.